Okay, welcome to Let's Get to the Marks. We're looking at A-level biology organelles. This is the Edexcel exam pack, Voice of the Genome. You'll find the link to this. It's a physics and maths tutor exam pack, brilliant website. I suggest you use it to get your A star in A-level, and you'll find the links to this in the description. All right, bacterial DNA is circular and surrounded by nuclear membrane. Now, we've got no nuclear membrane, so we can get rid of any answer with nuclear membrane, as bacteria don't have a nucleus. And of course, their DNA is circular. You either have like a chromosomal loop or a plasmid. You don't have linear DNA. Okay, let's look at question 2. The photograph shows an electron micrograph of a mitochondrion from a liver cell. The magnification is times 20,000. Calculate the actual length. So you need to know this equation off by heart, and it's I am, and it's image size over actual times mag. Now, we need to measure the image size. So anytime you're getting your ruler out, and we're going to measure across here with our ruler, anytime you're getting your ruler out, you are measuring the image size. You're not going to be measuring the actual size. And it's about 11.6 centimeters. And so what we need to do here, we can keep it because it doesn't specify the unit. We can keep it in centimeters, or we can do this in millimeters, or if we like, we can convert to micrometers. I generally convert to micrometers as that's the more common one they ask for you to do in the question. So you times the millimeters by 1,000, it'd be 116,000 micrometers divided by the mag of 20,000. So for this question, it's really your choice, millimeters or micrometers. Right, let's check out question 3. DNA synthesis in bacterial cell cultures has been investigated. Describe how DNA is organized in a bacterial cell. I recommend using a highlighter when you're doing these questions just to highlight key points. So chromosomal DNA is not attached to histones. Okay, when you're in a bacteria, their chromosomes are not attached to those proteins called histones. They also contain tiny circles of DNA called plasmids. So that would definitely differentiate, and those would be in the cytoplasm of the bacteria. You don't really find them in the cytoplasm of eukaryotic cells. They also wouldn't have a nucleus, and also we would find DNA loose in the cytoplasm. So those would be some differences. Let's go for question 4. I recommend you pause the video and have a go at the question before watching me go through the walkthrough. Okay, so for this diagram, describe the roles of part B and C in the formation of and transport of extracellular enzymes. Well, B is the Golgi apparatus, sometimes known as the Golgi body or Golgi complex, mostly an Edexcel SNAB. It's known as the Golgi apparatus. Proteins are folded, or we should really say they're packaged and modified here. The folding of the proteins happens in the rough endoplasmic reticulum. That's over before the Golgi apparatus here. It's not part of B. Sometimes a sugar could be added to the protein in B, which is the Golgi apparatus, to form a glycoprotein. And then they bud off in vesicles, secretory vesicles specifically, and those vesicles fuse with the cell membrane and are released by exocytosis. Although the main things here that we want to talk about really are packaged, modified, Golgi apparatus, that's 2 marks, vesicles, and then fusing with the membrane, and that would get you your 4 marks specifically. All right, let's move on. Question 5. Glucosaminoglycans, oh my geez, GAGs, are the byproduct of chemical reactions inside cells. GAGs are broken down by enzymes inside lysosome cells. I'm not even going to bother reading that. In MPS 1 is a genetic condition that results in the buildup of GAGs inside cells. MPS 1 affects the production of enzyme G that breaks down GAGs inside lysosomes. All of that was a load of nonsense. The question just says, describe what happens to lysosomes once they've digested their contents. So what happens then is the lysosome fuses with the cell membrane. Its membrane becomes one with the cell membrane, and then it releases its products of digestion outside of the cell. And we call that exocytosis. All right, next question. Plasmodium falciparum is a single-celled eukaryotic organism. P. falciparum causes the disease malaria, blah, blah, blah. You can pause the video and read that if you like. Describe how scientists could have determined that P. falciparum, whatever it's called, is a eukaryotic organism. So basically, the whole point of this question is explain why it's a eukaryote and not a prokaryote. And so we just want to say, look, if you were looking at it, you'd be able to see DNA is in a nucleus, 1 mark, whereas a prokaryote has no nucleus. It would have 80S ribosomes instead of 70S, which you find in a prokaryote. You could say larger ribosomes in a eukaryote. The DNA in eukaryotes is associated with histones. The ones in prokaryotes have no histones. Histones are like these little circles I've drawn here. The chromosomes or DNA in eukaryotic cells is bound around these proteins. So what we're doing is we're comparing and really describing the differences. Eukaryotes contain membrane-bound organelles such as mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum, whereas prokaryotes do not contain membrane-bound organelles. And there's your 4 marks, guys. Hope you're doing well. Let's go. The diagram shows the features of a human liver cell. We got cytoplasm at 1, 2 looks like the nucleus, and 3 is a mitochondrion or mitochondria if you're doing plural. Which of the labeled features in the liver cell contain RNA? Well, you'd find out mRNA in the cytoplasm when it's on its journey from nucleus to ribosome for transcription, right? And we definitely find mRNA in the nucleus because that's where it's made in translation. But what about the mitochondria? I suggest you look up something called endosymbiosis. It's not on your syllabus, but it explains why mitochondria and chloroplasts have their own RNA or genetic code. So the answer to that one was D. 1, 2, 3. Right. Some cells produce lactate during anaerobic respiration, or liver cells do. Lactate travels in the blood to the liver. Liver cells can absorb lactate from the blood. Deduce what happens to lactate in these cells. The lactate's converted to pyruvate. Spelling mistake there. Whoopsie. Pyruvate can be used to make glucose for respiration. And that basically is it. Okay, next part to the question. During protein synthesis, 2 amino acids are joined together to form a dipeptide. So let's check out this diagram. They want us to draw the dipeptide. We got the carboxylic acid group and we got the amino group. And we know that it's a condensation reaction. We're going to have water formed here. So we're going to lose the OH of the carboxylic acid and we're going to lose 1H of the amine group. So we just need to redraw this. And make sure you put the double bond O vertical and the nitrogen with the hydrogen. They can be on the same side. People like just for visual looks to put them on different sides, but they can be on the same side. It's totally cool to have them on the same side, the NH and the CO. Okay, there we go. That's your dipeptide. And in the middle there, which I'm going to circle here, this would be your peptide link. Remember, you also get a water molecule formed during this reaction as it's condensation. And that's your peptide link in the middle, although that wasn't specified in the question. Question 8, electron microscopes have enabled scientists to view the ultrastructure of cells. That means the organelles and the small things inside. Which of the following structures are found in animal cells? So you have the amyloplast, the chloroplast, the mesosome, or the ribosome. Ribosome. Yeah. Well, the amyloplast does what it says on the tin. It makes amylopectin and amylose, which are types of starch. So it is an organelle for synthesizing starch found in plant cells. The chloroplast, you all know that from GCSE. The chloroplast contains chlorophyll and it's got thylakoids and the granum, blah, 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 blah. It's basically where photosynthesis happens. So once again, that is in plant cells, not in animal cells. The mesosome is a structure found in bacteria. It's a infolding of the cell membrane in bacteria. And the ribosome, of course, is found in plant cells and animal cells. Which of the following structures is found only in plant cells? Well, we just covered that. That's the amyloplast for making starch. Which of the following structures is found in both plant and animal cells? Well, you get the smooth endoplasmic reticulum. It's going to make lipids for your waxy cuticle in plants. It's going to make lipids for cell membranes in both types of cell and lipids for energy storage. So there we go. Your smooth endoplasmic reticulum. Right, let's move on. Okay, question 9, zooming in. Which of the following parts of the statement is true for eukaryotic and prokaryotic? Both possess ribosomes. That leaves us with A and C. Eukaryotic possess plasmids. Actually, some eukaryotic cells do have plasmids in their chloroplasts or mitochondria, but they definitely are in bacteria, so A is incorrect. So we're looking at C here. Prokaryotic cells do not possess endoplasmic reticulum. Remember, they don't have membrane-bound organelles. It's one of the differences between prokaryotic and eukaryotic cells. Let's move on. Question 10. Zooming out a little here. The red squirrel population on Brown Sea Island has been affected by a disease called prokaryotic organism. Okay, really, this question is saying which of the following contain prokaryotic organisms, and it's going to be archaea and bacteria. Obviously, bacteria are prokaryotes, but archaea, the ancient bacteria. Carl Woese discovered the differences in molecular phylogeny by looking at the protein structure of like cytochrome C and various other proteins. So yeah, thermophilic bacteria, they are known as archaea. Now the next question is another one of those. Describe the features that could be used to identify prokaryotes in the blood. So again, comparing eukaryotes and prokaryotes, general theme with a lot of the questions on this syllabus. DNA is not associated with histones. We've got no nucleus. You wouldn't see a nuclear membrane. There'd be no plasmids. You'd see no pili, which are little hairy things around the outside of bacteria. No flagella, which is the corkscrew-like tail for propelling them. And you wouldn't see any membrane-bound organelles. And you can name one of those organelles. So mitochondria, rough endoplasmic reticulum, etc. Okay, question 11. The diagram shows some of the cell organelles involved in the formation of extracellular organisms. Name the parts of A, B, and C. Well, you can see that we got the RER up there because you can see the little ribosome stuck to the membrane of the RER, which is a fluid-filled sac. However, it's also attached to the nucleus, so that's a nuclear pore at A. That's where the ribosomes, that's where the mRNA will come out and leave the nucleus. So we've got a nuclear pore. Now, B's are Golgi apparatus. You can spot them by the little vesicles attaching and budding off at the ends. And then, of course, C must be a vesicle, or it could be a lysosome if it's containing lytic or digestive enzymes. Okay, cells can be observed using an electron microscope and a light microscope. The electron microscope shows an organelle in a eukaryotic cell. This, my friends, is a centriole, and the centriole is involved in the formation of the spindle organization. I know I'm going fast, people. Just make sure you pause the video every time I get to a new question, and you have a go. An electron microscope can be used to identify organelles in eukaryotic cells. Label the nucleolus. So here is the nucleolus. And the nucleolus is not found in prokaryotic cells because why can't we find a nucleolus in a prokaryotic cell? Well, they don't have a nucleus, so you won't have a nucleolus. By the way, that nice smooth black circle in the corner is probably an artifact or looking through the microscope. Explain why the nucleus cannot be observed at the end of the prophase in a eukaryotic cell. What happens to the nuclear membrane is that it breaks down or dissolves in the prophase. So we're just left with the chromosomes, and the chromosomes, they condense, okay? So really your 2 marks here is for saying that the nuclear envelope breaks down and the DNA condenses into separate chromosomes. Right, question 14, pause the video, guys, have a read of this. I'm just going to get on with it. So give 2 functions of the Golgi apparatus. The functions of the Golgi apparatus, we know that is for packaging and modifying proteins. You could give an example, adding a sugar to a protein to make a glycoprotein, but it's also for formation of vesicles, secretory vesicles. Okay, a 3-phase protocol. Pause the video for this next question. The 3-phase protocol, what's it for? To do with testing drugs. Well, phase 1 sees if the drug is harmful, also tests what's the safe dose. Phase 2, I can't spell properly. Whoopsie. Yeah. So phase 2 is about efficacy. Does the drug work? Is it effective? Now that we know it's not toxic or seriously toxic, and we know the safe dose, is it effective? And then phase 3 is where you look at the data, you analyze the data, and you study whether your results, whether you have a positive correlation or causal effect in the results, and you use statistical tests to check the data. Okay. Now you also might look for rare side effects. Right. Question 15, pause the video, have a read, have a go. Right, some fish basically live in the Arctic and they have this antifreeze glycoprotein inside them, AFGP. Describe how this polypeptide is then processed to make the AFGP. Now we're told it's a glycoprotein. Okay, so we know what's going to happen in the Golgi. So first it's translated on the ribosome in the RER, right? It gets translated. If we want, we can talk about the folding that happens there. You know, it becomes a beta pleated sheet or an alpha coil, depending on the hydrogen bonding. And we can talk about the secondary, and we can talk about the tertiary structure. The interaction between our groups forms the tertiary structure. That all happens on the rough endoplasmic reticulum. Then it is transferred to the Golgi apparatus where it is packaged or modified. You'll get a mark for that. And you can say it's secreted in a vesicle and moves around the cell, but you could have also said a sugar is added because they told us it's a glycoprotein. So we could have added that a sugar is added to make a glycoprotein, and that would happen in the Golgi. Right, pause the video for question 16. All right, let's check this question out. Protein synthesis in eukaryotic cells occurs in, it's going to be ATS ribosomes and they are found on the endoplasmic reticulum. You don't find ribosomes in the nucleus. Awesome. Although you do find the material for making ribosomes, like the nucleolus is where they're made. So yeah, I'm waffling. Many of the proteins synthesized become extracellular enzymes. Describe what happens to these proteins. Oh my gosh, we've done this question like 50 times, worded in different ways. So it's passed through the RER, the rough endoplasmic reticulum, to the Golgi apparatus or Golgi body. Okay, if we want, we can talk about at the RER, the protein structure is decided. So it gets a secondary structure with the hydrogen bonding, it becomes alpha or beta, and then it gets the tertiary structure, the 3D shape. It's then sent to the Golgi apparatus through the RER, where it is packaged and modified. This could include adding a sugar or a carbohydrate, you could call it, or a glucose molecule to make a glycoprotein. Okay, you could add a lipid to make lipoproteins. And then it leaves via a secretory vesicle. That secretory vesicle, if they're talking about extracellular proteins, if they mention that, they're giving you a hint that they want you to say, hey, the vesicle fuses with the membrane and releases the contents by exocytosis. And this question said, how's it released from the cell? So that's always worth putting. Cells in people with these diseases produce incorrectly folded enzyme molecules. Explain why enzymes that are incorrectly folded cannot carry out their function. So if you're not folded correctly, well, how does that happen? That happens, your folding is based on your interaction between the R groups. That gives you your tertiary structure. So if we have some sort of disruption to the R groups, then really that's going to affect our tertiary structure, our 3D shape. It means our active site will no longer be complementary to the substrate. So active site is no longer complementary to the substrate. Less enzyme substrate complexes form. Therefore, they cannot carry out their function. Lovely jubbly. Of course, the function here of the enzyme is to catalyze a reaction, and obviously it can't do that if it has the wrong 3D shape. So question 18, in prokaryotic cells, ribosomes are smaller than ribosomes in eukaryotic cells. That's right. Prokaryotic cells have 70S ribosomes, eukaryotic have 80S. Okay, we've got a picture little here of a unfinished picture of a prokaryote. Okay, it's a bacterial cell. Complete the diagram to show the cell membrane, a mesosome, and a plasmid. Right, a mesosome is like an infolding in the cell membrane, so that the membrane kind of ducts inwards, and it's got various bits inside that little bit, the mesosome, where it infolds. The cell membrane, you want a line drawn inside the cell wall, because obviously the cell membrane comes before the cell wall, and the plasmid is just a tiny circle of DNA. Right, question 20, zooming in. Some bacteria have a capsule that is located, it's going to be outside the cell wall. The capsule, glycocalyx it's made of, it's like a snotty slime mucus layer, is around the outside of bacteria, and that's the capsule. So it's going to be outside of the cell wall. Okie dokie, a newborn baby. Pause the video for this one, guys. This is question 21, and explain these results. All right, pause the video and have a read through. Okay, we're looking at a drug called interferon, and what does it appear like interferon does? Well, as the level of interferon increases, the survival time for the mice also increases. 1 mark. Boom. So it looks like this drug interferon inhibits the replication of the virus. So the virus cannot replicate. Yeah. How does that affect the virus? Well, if it can't replicate because that's being interrupted or inhibited, then the virus can't spread so much. It can't spread around the cell, can't burst the cell, it can't spread to other cells. So you're going to reduce the spread of the virus throughout the body. And that's because you've interrupted the replication of the virus. You could also point out that there's a positive correlation between the dose of interferon and the survival time of the mice, if you don't want to say the first line. All right, let's move on. Pause the video, guys. Have a go. Right, here's the answer. Explain why interferon made by genetically modified bacteria is different from interferon made by animal cells. So think about interferon, it's a protein, right? Um, so what's the difference in the construction of proteins when they're made in a bacteria compared to when they're made in a eukaryotic organism? Well, bacteria don't have an RER, so they're not going to have the same type of folding, the same level of folding and tertiary, secondary, and all of that business structures that we have in eukaryotic proteins. Okay, they also don't have Golgi apparatus for modifying the proteins, so they can't modify and package proteins in the same way as eukaryotic cells. So they're the two big differences between how bacteria and how eukaryotes make protein. Glycoproteins made in animal cells are released in the extracellular fluid, and that's by exocytosis. Cytosis means cytoplasm, exo means outside the cytoplasm. So name the structure labeled C. Now you'd be forgiven for going for middle lamellae because it's in between two plant cells, between the plant cell wall. You might say boarded pit, but it's actually a plasmodesmata. And this is the little channel that connects the cytoplasm of the two cells and allows for exchange of materials or the transport materials by diffusion, by osmosis between the two cells. And so that's what plasmodesmata are for. They connect the cytoplasm, so it's like one interconnected flowing cytoplasm between all the plant cells. And it also allows for communication. They don't say, hello, how are you? But they send little proteins which have specific shapes. Yep. Yeah. If you want me to do more of the videos, let me know.
You're still here? It's over.